Dr. Dunn is a best-selling author of several books, most notably The Man Who Touched His Own Heart and A Natural History of the Future, What the Laws of Biology Tell Us About the Destiny of the Human Species. And he's written several science essays in numerous different journals, which we talked before. He earned his PhD in Ecology and Evolution at the University of Connecticut in 2003, and currently is professor at North Carolina State University in the Department of Applied Ecology, where he runs the yourwildlife.org program. He's already given us an amazing talk, and I will have to say, I thought whoever baked the rolls today had very good flavor to them. I really appreciated that. And it's my immense honor to be able to reintroduce back up to the stage Dr. Dunn. Thank you.
Thank you again. Can you hear me? Is the mic on or not on? Let's see. All good? I'm just glad I'm not competing with the cannabis talk. So when I was asked what I would talk about at this meeting many months ago, I had the great idea that I would talk about themes that interlace between these two books. But I didn't think about too much what those themes would be, and then in the last couple of weeks I've actually had to figure out what that talk would look like. And so one of the things I've recognized that two themes that go throughout both these books, one is the importance of evolutionary history and thinking about diverse phenomena of human medicine and biology, and the other is the importance of recognizing the value of broader biodiversity. So these are themes that emerged in the first talk. They're a little more concrete here. And so I'm going to walk through in this talk sort of first that sort of evolutionary thinking component, and second that broader biodiversity component. First, for the biology of homes and what we understand about the biology of homes and how it's changed, and then later for the biology of hearts. And so if we think about the biology of homes, there's actually not been that much evolutionary study of how homes have changed through time. We know that homes, as we think about them as human homes, are relatively recent, and so too all of the species that live in those homes. The oldest homes ever documented to date are about 200,000 years old. And so that's a little far left on that, but probably homes emerged around 500,000 years ago, so built structures that humans would live in. Before that time, our understanding is that our ancestors almost certainly built nests. This is what chimps do, it's what bonobos do, it's what gorillas do. And so in thinking about what our biology of our homes is like, what the biology of our hospitals is like, what the biology of family practices is like, one of the first things I'm going to do today is to tell you a little bit about what we know about what lives in chimpanzee nests. And so when we started studying chimpanzee nests, our thought was that they would be dominated by chimp pathogens and chimp ectoparasites. And so full of weird worms, full of weird skin microbes, but nobody had ever studied it, so we didn't know. And so my grad student, Megan Temis, teamed up with a group working in Western Tanzania to actually go and sample chimpanzee nests and see which species are living there, to give us a kind of baseline of what our ancestral homes would have been like in terms of their composition of microbes. What Megan found is that the chimp nests were basically entirely dominated by soil microbes and leaf microbes, and there was no sign of chimp-associated skin microbes, fecal microbes, and ectoparasites. And our interpretation of this is that this is likely what our ancestral homes look like. They were really soil and leaf. And part of why this happens is that chimps, like gorillas, like orangutans, rebuild their nests every night. And so there's really not the opportunity for bodily associated things to build up. This seems very esoteric, but as we start to study human homes, it becomes clear that it's very relevant to how we think about our own biology. And when I started studying human homes in 2010 or so, there were actually very few holistic studies of what would live in a room like this one. People had done culture-based work looking for individual pathogens, but we didn't have a broad sense. And in part it was because biologists like me, ecologists trained in doing field work, when they would go and look at a landscape like this one in Manhattan, they would zoom in on those green habitats. They'd look at forests, they'd look at Central Park. They wouldn't study this gray landscape. And so we started studying this gray landscape to figure out what lives in our modern homes. This is Matt Bertone. He led a lot of this work. This is not to scale. He's a normal-sized human. But so we first started by looking not at microbes, but at arthropods, insects and their relatives. And one of the things that we were told by everybody who worked in homes is that when we did this, we were going to see these few problem species. That's what we'd find in homes, but there really weren't any studies. And so we went into homes, and what we found was all sorts of arthropods everywhere we looked. There were arthropods living in each little habitat of the home. And in fact, in the first 40 homes, we found more than 1,000 species of arthropods. And a large proportion of them were house-dependent. They essentially moved into Mesopotamian houses, they evolved to take advantage of houses, and they spread with humans around the world. So much so that by the 1600s in Boston, when no fewer than 40 household beetle species had already been introduced from Europe to those houses. And so there's a spread of things that depend on us that we don't understand very well. Some live off our garbage, our decomposing things, our foods. A lot of those species eat those species. And so this was a surprise, and it got us more interested in what was living in these houses more generally. This was very much an exploratory phase of this work. And so what we did is we had people sample dust in their houses, and then we could kind of do like a CSI dust, CSI bed, bathroom, and beyond. And we could decode the DNA that was present in that dust. We'd make more copies of it. We see who was present. And we engaged people all across North America to send us the dust from their homes so we could see what was living in that dust. We looked like Santa's workshop for a lot of the time when we were doing this. What did we find? We didn't know what to expect. We expected tens of species. We expected food-associated pathogens we know about. We expected, some bread mold. So we found thousands of species of plant species, that were characteristic of what people were eating. And so, for example, if you're smoking weed in your house, we can detect it in the dust just by the little bits of plant DNA that float all through the house. I'm not accusing, but Brian, if it's there. We found 40,000 fungal species in homes. That's more fungal species than there are named fungal species in North America. We found 2,000 arthropod species based on DNA, 80,000 bacteria species. No one's ever studied the protists in homes. And so there's a lot going on. And so this was a surprise. The other thing we found was that the species living in homes were rather unique. And so one of the groups of species we documented were species that really liked the extreme environments that we found. And so, for example, hot water heaters have a lineage of bacteria that's known from hot water heaters and hot springs. We don't know how they get there, but they're in most hot water heaters. It's the same species that we use for the enzyme Taq polymerase that we use for almost all modern DNA work comes from these microbes. They're living in our houses. Fridges have a lot of cold-tolerant microbes that we see in the Arctic. Freezers have really extreme cold-tolerant microbes we see in the Arctic. Fermented foods have lots of acid-tolerant things. The soap dish and dishwashers has extremely tolerant fungi that have never been documented anywhere else on Earth. One of the kinds of fungi species in soap dishes and dishwashers is known from the feces of tropical fruit bats and from dishwashers, which mostly says we've not explored it yet. We don't understand these connections. We don't understand where these species are coming from. And then inside salt crystals, we find halophilic, salt-loving bacteria that are from wherever the salt was gathered. I was just talking to one of our helpers with the video and with the talks today, and he was saying that he, at home in Brazil, he cures meat using a special salt that his family thinks is really great for curing meat. It's very possible that that salt is actually offering specific microbes to that meat curing. And so it's all over homes. There's a lot going on. But it's very clear what's living in homes is very different from what our ancestors were exposed to. And to the extent that we're controlling what's in these environments, our control is modest, and with the exception of hand washing and clean drinking water and basic hygiene that prevents pathogens from establishing, a lot of this control is probably doing things that aren't great. And so, for example, this is a plot where each point is a sample of a house, and points are closer together if they're more similar in composition. And so one thing you can see is that there are these green points that are associated with food. And so these are samples that have food-associated microbes on them. That purple dot in the middle, that's somebody who had a hot dog on their, they got mad, threw a hot dog at their TV, something like that. The blue dots are outdoor associated leaf and soil microbes that are coming into houses through doors and windows. And so there's sort of this is one way we're modulating which microbes are present, by opening doors, by opening windows. And then the pink and the whatever color that is, sort of a burnt umber maybe, that's pillowcases and toilet seats, which we can't tell statistically apart. And they're mostly just a function of that where we put our bodies, we put our body microbes. And so this, whatever's happening here, it's not a fine-tuned kind of control. The other thing that we do that has a big effect on which microbes we're finding in houses is who lives in the house. So the more people live in the house, the more body microbes you see in a house. The more men live in a house, the more armpit microbes you see in the house. The more women live in the house, the more vaginal microbes you see in the house. And we detect this just in the air around the house. If you have a dog, and I just Googled strange-looking dog, this is what came up. It was useful for this moment. Dogs have a huge effect on which microbes are in the house, and there are a whole suite of dog-associated microbes that become much more common in the air in houses with dogs. Some of these are probably beneficial, some are not, but it's a big effect. But it's not really control. The other thing we know is that closing windows, opening windows, favors different microbes. And so closing windows rules out the outdoor microbes and tends to favor body microbes. Opening windows does the reverse. This has been shown in a hospital context. If you open the hospital windows, you actually get more leaf and soil-associated microbes in hospital rooms. And because those compete with pathogens, you see fewer pathogens than in a recirculating air situation. We also know the AC and heat units favor specific fungi that are pretty weird fungi. When you turn on your AC unit and there's that smell initially, that's essentially the fungus in the AC unit exhaling. And then we also know that when we chlorinate water systems, which is where we don't have clean drinking water supplies, where our aquifers are polluted, we have no other approach, but it favors chlorine-tolerant microbes. And so we have this kind of control, but it's not a sophisticated kind of control. And the net effect is when we look at homes versus bodies, homes are really dominated by body-associated microbes, which is not the ancestral condition our bodies evolved in. And this is even more extreme for hospitals and probably most clinical settings. Add to this the reality that we spend 95% of our time indoors. Think about your own day today. And our exposures are really radically different from what they would have been historically. And so from lives outdoors amidst soil and leaf microbes to lives indoors amidst body microbes, and this is a very fast transition. The net effects of this is that the microbial composition in houses today appears to predispose people to allergy, asthma, and other inflammatory diseases as much via what's present as what is absent. We're still beginning to understand this. It's clear there's a link. The link, its causal mechanisms are not as clear. But it's clear that something like this is happening, that there are all these microbes from soil and leaf that we used to be exposed to that had immunomodulatory effects, and now we're being exposed to our own body microbes constantly. I could talk more about this later. We're beginning to see experimental projects that aim to change household microbes to benefit people in terms of allergy and asthma. Some are effective, some are not. We don't totally understand which are which, but clearly there's something going on there. We've also created an environment that promotes survival of biocide-resistant bacteria and opportunistic pathogens, which are often poor competitors. And so we see this in chicken slaughtering facilities that are bleached daily, and the floor is basically covered with not very competitive microbes that have these cassettes that are full of antimicrobial resistance genes. And they're not microbes that do well in nature, but when you bleach everything all the time, they do great on those floors. We also see this in some hand sanitizers. We're seeing a weird pseudomonas that actually like to live on the hand sanitizers because they don't have much competition and they can tolerate some of the antimicrobial compounds. And then we're seeing this in shower heads, where some of the species that do well with lots of chlorination actually have resistance genes. And so we're not doing great things with regard to which species we're favoring, and often really simple interventions like opening the window create better environments. The added piece of this is that in an environment where beneficial microbes are not acquired during birth, they're very unlikely to be acquired later on. And so one of the things we see with C-section babies is a lot of variability in their later health. And we know there are negative average effects of C-section births, but some people acquire all the gut microbes they need. It looks at some of those are coming from dogs later in life, and we're seeing C-section babies who actually have dog gut microbiome species that they're acquiring. And so we've created this weird condition. It's not clear how we fix it, but it's sort of a modern piece of our story. And this is a great paper that covers some of this, the C-section birth gut microbiome work that you can refer to later if you're interested. And so in trying to keep life out, we favored a set of species that have adapted to indoors. We've excluded beneficial species to our own detriment in the context of allergy, asthma, autoimmune disorders. And we've created homes that are less healthy than those built by our ancestors 8 million years ago, all of which I think is interesting. And so if this is not the future, then what? And when I was here before, which I thought was 2017, when I was thinking about this in the plane, turns out it was 2013, I talked about fecal transplants, and at that time they were kind of laughable. They got a little giggle. Now they're not laughable. And I talked about fecal transplants and what they would be for the future. And now the fecal transplant landscape has really changed. We have direct from donor transplants. We have processing, which we didn't used to have. We have pills versus liquid. And we also have specific species that are being offered. I think as we look to houses, my prediction is if I come back in 5 to 10 years, what we're going to see is an initial focus on reintroduction of outdoor microbes into homes, akin to the earliest fecal transplants, initial haphazard results, one thread of future interventions that will focus on bringing microbial processes into the homes, another thread that will focus on specific microbes used as therapeutics, and all of this is already happening in sort of niche and marginal contexts. But I predict if I come back, this will be much more mainstream, and we'll be struggling with understanding what's working and what's not working. Now, that's an evolutionary context and aspects of the home. What about the heart? Can I pull these pieces together? With the heart, I'm going to focus on atherosclerosis, which was first discovered almost certainly by Leonardo da Vinci. I read about this a little in the book. Leonardo da Vinci did lots of dissections. He loved doing dissections. And more than once, he would sit next to somebody who was really old with the sense that maybe he was waiting for that person to die so he could do a quick dissection. And he did lots of work on human hearts and non-human hearts. But one of the really most important dissections he did was there was a very old man who he was sitting next to who then died. And he quickly, after the man died, found some place to do a dissection of the man. And what he found is that he had twisted, clogged arteries that Leonardo thought were the cause of death, and he thought they were associated with a good death. As a well-spent day brings happy sleep, so life well used brings happy death. We can talk about that change of perspective. But he was recognizing atherosclerosis, but what he found was not built upon because most of his scientific work was lost for hundreds of years. And so it was much later, and so here's one of his drawings. He talks about tortuous arteries. It's much later that people start thinking about atherosclerosis in humans. And one of the things that happens as people start thinking about atherosclerosis in humans is it's very medically oriented. And so there's almost no study of what atherosclerosis looks like in other species. Very, very little. And so the general assumption is because the hearts of chimps and humans are very similar, that the problems are probably very similar. We're so like each other. Our hearts are so like each other. And even there are some cases of chimp heart attacks that suggest maybe something similar is going on. But it wasn't until very recently that Nissi Varkey started to actually document some of those heart attacks in chimps and look at what had happened in the heart. And when she did, what she found was, first of all, there was no evidence of atherosclerosis in any of those chimps that died of heart attacks. Second, the chimps had some different kind of heart problem caused by fibrosis, which you see stained in the blue, that nobody had documented before, and it's not clear what's happening. And so this then raises the question, is what's going on in humans that's not going on in chimps? Why are they different? And the first possibility is that the chimps were fed a less sort of atherosclerosis-prone diet. Clearly not the case. These chimps were being provided, in many cases, food that was not very good. Maybe they were eating less meat, very little meat in the diets. Maybe that's part of it. The second thought is maybe cholesterol is doing something different, but chimp cholesterol levels and human cholesterol levels look very similar. Doesn't appear to be differences in cholesterol. The third thought is, well, maybe it's like something specific about human history and diet, but if you look at different groups of humans, all humans studied to date, whether ancient humans or modern humans, seem to be more prone to atherosclerosis than do chimps. So something is still missing. And I'll just quickly tell you what appears to be missing is that there's a change in sialic acid in humans versus chimps that evolved about 12,000 years ago. And so in chimps, both of these forms of sialic acid are present, the NEU5AC and NEU5GC. And there's an enzyme that converts one to the other. In humans about 12,000 years ago, the genes for that enzyme broke. And it looks as though the reason they broke is that ancient malaria bound to this sialic acid on the right. And so humans escaped malaria by evolving this to have just this one kind of sialic acid. Now, this was a very short-lived break, so that's the malaria binding site right there. And it was a short-lived break because then later, another kind of malaria colonized humans from gorillas that evolved the ability to bind to the other sialic acid. And so there was a moment we didn't suffer malaria, didn't last long. Nature hates a healthy human. And what's interesting about all of this is that Nissi Varki and Ajit Varki have now shown that when we eat red meat and some other kinds of meat, that the sialic acid from that meat, which includes the one on the right that I showed you, becomes incorporated into our cells and generates an immune response in our bodies. And so it looks like part of what's going on with atherosclerosis is that the body is actually reacting to that non-human meat sialic acid that becomes incorporated into our cells. And in general, humans seem to also have a more inflammatory condition, and those two things together seem to be associated with atherosclerosis. And human-like mice that produce the same sialic acids that we do also suffer from atherosclerosis if they're fed meat. And so it seems like this is part of the story of atherosclerosis. It's a bigger story. But my main point today is by considering the evolutionary part of this story, we see different questions that didn't emerge before. Then there are all sorts of questions about how we respond to this, what else is going on. We can talk about them later. But the big point is that the questions change. We still have no idea what's happening in chimp hearts when they develop this fibrosis. No one's ever studied it. Now to come back to this broader biodiversity phenomenon, when I started working in houses, one of the things that always happened is if I'd come to talk to people about the life in their houses, and people would say, well, what good is it? And as a biologist, I hated this question because I thought these species were just interesting. You know, they were parts of broader nature. I just liked them. But finally, we started to listen to this question. And so we thought, okay, how do we think about the biology of these species and what use they might have? And the first one we thought about was camel crickets, which are really cave crickets that moved into houses. And so we thought, well, we know that they eat really hard to break down foods. And so what if they have gut microbes for breaking down really recalcitrant materials that we could use industrially to serve humans? And so I teamed up with Amy Grendon, who works on something called black liquor, which is the waste product of the paper industry. It's lignin, so the hard stuff from trees, and an alkaline bath, and right now it either gets burned or dumped into rivers. What she didn't tell me is that nobody had ever found a microbe that could deal with this stuff. And so we tried the camel crickets, and the first camel cricket, the first individual camel cricket we looked at, doubled the number of bacteria species on Earth known to be able to break down lignin, and included species that could break down lignin in this alkaline bath, and that can turn this waste product from the paper and pulp industry into energy. And so this seemed like a good omen for thinking about broader projects. And about the same time, somebody called me and said, Rob, we're going to have a beer festival. Do you have any yeast from your work we could use to make a new kind of beer? I thought about this a little bit. Ann Madden at the time was working in my lab, and Ann worked on paper wasp nests. And she knew that the microbes in paper wasps often include yeast. And so Ann looked to an individual paper wasp from her house, sampled it. The first wasp we looked at had a new kind of yeast that we could use to make a sour beer in a month that would typically take a year to make. And that's now been commercialized. It was very popular, that's Ann. And we've now found that those same yeast can be used to make new kinds of breads. We also then started thinking, what about these cold-tolerant microbes we're finding in refrigerators or in cabinets? And what about the things we find on fish skin or more upmarket in caviar? We looked to fish skin, we looked to caviar, and we found yeasts that can help to control the process of decay in these foods and slow decay. Interestingly, the same microbes we found in these two places are the microbes that were in the body of Ötzi the Iceman and may have helped to preserve him through time, which doesn't mean I would like to taste him, but it just gives you a broader sense. And then sometimes we start these projects and we start in one place and they go another direction. And so we're really interested in the exoskeletons of household ants, which we know those exoskeletons produce antimicrobials. Could we learn from them in new ways? And so we teamed up with people in textile and designs to try to imagine making a new kind of clothing that worked like the outside of an ant. That didn't work, but the people in designs noticed that the surface sculpturing of the ants was actually beautiful in a design context and started to make new patterns for pillows and other things that you can now buy based on the ants. No medical solution here, but maybe just some interesting design space. But we also found that by studying other ants, we could find antimicrobials from those ants that were functional against some resistant pathogens, and so we're now working on those. And so these are just species in our backyards. Face mites, another example. We've started working on this. We're not very far, but we know that in most contexts, people have hundreds of thousands of face mites and don't generate an immune response. That suggests to me that the face mites are actually producing immunosuppressive compounds. And so one of the things we want to do is to check these face mites for useful immunosuppressive compounds that we might use in a medical context. We're not there yet. And then as we look to nature more generally, many of these species in our household environments prove useful. For example, we've now found species in tap water coming out of these water fountains at schools that precipitate gold from the tap water streams and from waste streams, and I've been told that could be useful. Bringing this back to the heart, what's the broader value of biodiversity in thinking about hearts? It turns out there are lots of values here. And to talk about this, I need to talk just a little bit about heart transplantation, which starts as this sort of macho-filled endeavor to do this new, bold thing, but which also is clearly going to help humanity in the long run. And so there were as many failures early on as there were successes. Early transplants of chimp heart into a human, transplants of human hearts very early on in our understanding into other humans, which the first transplant patient lived from Christian Barnard's work. Christian Barnard became very famous, but then the first transplant patient died 18 days later. And so there's this early struggle to make this work, and clearly one of the things that was key to making this work was immunosuppressive compounds. And so I think one of the real heroes in the history of heart transplantation is cyclosporine. What I think is less recognized is that cyclosporine originally comes from a soil microbe. What's even less recognized is the natural history of that microbe and why it works. It was found in the soil, but it's not really a soil microbe. As was discovered by Kathy Hodge recently at Cornell University, it's actually a microbe that takes over the bodies of some beetles, keeps them alive, relies on their body to get more food, and then grows from their body into animal feces that those beetles crawl into. And so the active compound, cyclosporine, that this fungus is producing is partially being used by the fungus to suppress the immune systems of these insects. And it looks like part of how it works in humans is that it's suppressing such ancient parts of the immune system that it's functional both in insects and in humans. Why this is relevant is that today we've studied maybe 10 of these fungi and their relatives for their potential medical value. There are tens of thousands of these fungi that no one's ever studied. And so the specific story suggests the broader potential. And this is one of these fungi that infects ants, causes them to crawl high up on a branch, where it then grows out of their head and produces these fruiting bodies that it wants to be eaten by birds, that birds then disperse. For this fungus in particular, no one studied its medical value yet. I suspect it has good values. The other interesting side note here that I hadn't thought about until writing this talk is, and I noticed in a recent paper, is it's now been argued that one of the reasons some of these first transplants didn't work is because of the sialic acid differences between the chimp hearts, later the baboon hearts, and now pig hearts, and human hearts. And if that's right, it's really important because there's this big move now to use pig hearts in transplantation. But if the sialic acid is part of why this hasn't worked yet in humans, it's something we need to be tending to, and we could tend to. It might not be, but it's interesting to at least think about. And then one other example here that I really love is that of the Japanese scientist Akira Endo. And Akira Endo grew up in post-war Japan. He grew up very poor. He grew up gathering fungi with his grandfather. And he grew up fascinated by the power of fungi to be dangerous and delicious, and became very interested in using fungi. And the first way he used fungi was to do sort of industrial food technology, to clear up pectin from some kinds of ciders. But then he went to the US and he saw all the problems with heart disease in the US and all the cholesterol-associated problems with heart disease. And he thought, if I could somehow interrupt the cholesterol pathway in humans, I might be able to produce really something very valuable to society, and mostly to US society, because he saw the problem as being a US one, which is interesting historically. And he knew that some fungi fought other fungi by interrupting their ability to produce cholesterol. And he thought if he looked to enough fungi, he would find some that might be able to do this in humans. And so he screened fungal strains one by one for their ability to interrupt this pathway. This took years of his life. When his bosses progressively got angrier and angrier, he screened 6,000 fungal strains. In 1971, he finally found one that produced a compound called citrinin that works in rats but causes kidney stones. In 1973, after his bosses told him they were going to fire him if he didn't stop, he found one called compactin that worked in petri dishes but not in rats, and then he was done. But he went home and he talked to a friend who had a bunch of chickens, and he tried compactin in chickens. It worked in chickens, and that was enough of a success to get him a little bit of headway and produce the ancestor of the compound we now know as statins. And so statins start with this biodiversity discovery. They have all of this happenstance, and we still don't fully understand why statins work in some ways in one person versus another person, much less in chickens versus rats. A paper came out today that's worth checking out that suggests some of the effectiveness of statins is modulated by the gut microbiome. And I can talk about that more later. I just read it this morning, so I can talk about it briefly. But this, along with other clinical interventions, have amazing effects on deaths from cardiac heart disease. Just an extraordinary success story, and because of biodiversity in some real way. And one final example of the value of biodiversity and evolutionary biology together is work that's thought about comparatively considering the hearts of different animals and how they work. This is a baby blue whale heart, no longer attached to the blue whale, obviously. And one of the things we know about the hearts of different animals, they vary radically, but their heart rates also vary radically. And so it was noticed in the 1940s and 50s that there's a relationship between heartbeats per minute beating and life expectancy across animals. And one of the things that people recognized in seeing this relationship was that if you looked at it and you could find species that did different things, if they got more heartbeats than they should expect, something interesting biologically must be happening, and so maybe something we could learn from. The other thing that we in our lab have found is that one cause of variation among species and how many heartbeats they get is sociality. So social species like primates get more years than they should otherwise, in part from caring for each other and sort of extending lives in that way. But to go back to those beasts the beasts we want to understand, one of the animals that gets way more heartbeats than it should is this Arctic ground squirrel. And it appears to be that part of how it does it is by basically reducing its heart rate for part of the year when it hibernates. And so there's been lots of work starting in the 1950s to try to figure out what it's doing. It's now clear that part of what it's doing is it's producing adenosine, and adenosine slows the heart rate down and allows the animal to go into hibernation. What some of you might already know clinically is we also now use adenosine in clinical context to alter heart rate. And so here's another example of where this comparative work is interesting. I think the other example is that we know that we as humans get about 1.5 times as many heartbeats today as we should predict based on our heart rate, and that's thanks to people like you and the work that you've done to move us fundamentally up from this line and to extend the lives of so many people. I'll close now by just saying, as we think about evolution and biodiversity, the truth is we've barely started. And we barely started in the sense that most of the species on Earth haven't been named. The estimate right now is that there are 1.5 million beetle species on Earth, 5 million insect species in general, 7 million arthropods in general. 80% of those are undiscovered, not yet named, not yet studied in any way. If we look at bacteria, for a long time, nobody's even tried to estimate how many bacteria species there might be. The most recent estimate suggests there may be 1 trillion bacteria species. If it's off by 3 zeros, we're still at 1 billion. Just 30,000 of those are named. And so we're clearly just starting to explore biodiversity, and we can only begin to appreciate the values of biodiversity once we name those species and begin to study them. And then we have things like bacteriophages. We think that most bacteria species have their own viruses, their own bacteriophages, and some have several. It's possible if there are 1 trillion species of bacteria, there are even more species of bacteriophage. And so this work is really just beginning. And what I've tried to show you is the value this kind of work can have by twinning and intertwining these two stories. But the truth is, this is a much broader endeavor to bring these insights eventually to clinical application, which often takes a long, long time. But I think there's great hope in what we're going to be able to do, and we've become much better at being able to do this quickly. And so I'm hopeful for the future of what biodiversity and evolution can offer us and what's next. And so I'll close there. Thank you.
All right, if anybody has any questions for Dr. Dunn, come up to the microphones. I'm going to start off from one from our last lecture where it was online question that we didn't have a chance to go over. But that question was if you can speak to the probiotic Akkermansia, which kind of has shown some evidence in obesity. And I don't know if you have any thoughts on that.
Yeah, so Akkermansia has got an interesting story. I'll try to be more pithy than this morning. It's not my nature. So 8 years ago, it looked like Akkermansia was the key microbe for the human gut lining. We didn't pick it up in fecal samples because it's so intimately associated with the mucin and the gut lining. We know that when it's missing, there's a much more pro-inflammatory condition that's created. And my understanding is that probiotics of Akkermansia, if they can become established, have a lot of potential. But the other thing we've come to understand is that Akkermansia is really a microbe of Western industrial cultures, that other cultures and probably Western populations historically, there were totally different microbes lining the gut. And so it looks like given our modern microbiomes, it may be a functional way to restore, essentially makes the gut lining more solid. It reduces the permeability. I don't know the studies looking at under what conditions it's most likely to establish, which would be the key piece of this. But it's also worth recognizing that it's a kind of fix it given where we are. It's not taking our gut microbiomes back to what they would have looked like even a few hundred years ago. But it's a very interesting one. And our perspective has really fundamentally changed about it. Thanks for that question for online person.
All right, we got a chance for some more questions from the audience. I know you guys are very inquisitive, hopefully not in a food coma. So let's see, we got one up here.
Hi, my question is, a lot of your talk is about how things that we do are affecting our microbiome and our holobiome and genome in ways that we don't anticipate. With these discoveries of if we incorporate this bacteria or this substance from this bacteria, it has these effects, what are the potential long-term risks that we're not anticipating of changing it in those ways?
Yeah, so that's a great question. Can everybody hear that? So what are the long-term consequences of messing with our microbiome in another way? And the temptation is for simple fixes, and some of them have proven clinically successful. So first they've shown hope in rats and mice, super different gut microbiomes. A subset have been shown to be hopeful clinically. But typically the time horizon of those studies is very short, and it doesn't include a full human life, and it doesn't include evolutionary changes. And so I think often we don't know, and the trick is, I think this is the challenge with the microbiome medically. You have 1,000 species in your gut. It's a super dynamic system. We don't have great ways for thinking about that many moving parts. And so I think that next years are going to tell what those look like. I mean, I would watch, there are now transgenic gut microbes that are moving toward the market. Those microbes continue to evolve. And so I think this is one of these things to watch. How do they evolve in the human body? How do they change? We're giving them new special attributes. Do they keep those? Do they shed them? Do they pick up other sort of problematic attributes? And the trick is, those consequences are so long-term, we're not going to see them maybe for 10 years, 20 years, 30 years, 40 years. And how do we remain risk-averse enough to keep patients healthy? Again, I'm not a clinician. While also recognizing there are tons of microbiome problems right now that people are really suffering from. And because it's such a different kind of challenge, I don't think our old ways of thinking about risk and communicating risk, to go back to your talk, are necessarily going to be the best way to do this. And so I think we need to be having new conversations. And in a way, some of these applications are moving faster than those conversations, in part because gastroenterologists, for example, are so desperate often for things that will help their patients. So I think it's tricky, and we just don't have a good way with this complexity. And I could keep talking, but I'll stop there. Thank you.
All right, I'm gonna get my money's worth. I love the stories of the soil microbe that gave us this logical answer of it does this in nature, therefore maybe it can do this in our body. The way that drug discovery works on scale right now is we have libraries of hundreds of thousands of molecules that we just kind of throw in a dish and see which ones have effects. How do you see kind of the commercial drug discovery incorporating with the work that your field is doing?
So I think we need both in concert, and I can talk about it in the food context. In the food context, the very same thing happens. Novozymes has a giant collection of fungi and bacteria they've collected. You want to make a new food, they go to their freezer, they pull out this futuristic vat of microbes, and they try each one. The challenge is that often those collections are very narrow. And so what we recommend is that you combine that approach, that sort of screening one by one for the trait you want, with a kind of natural history, evolutionary guided search for something that might have value. And so we've found that we've been very successful at finding new microbes for food industry that have new attributes that are just missing from the collections people have. And we do it not by searching everything in nature, but by thinking, where might we find something like this? And so as an example that ties to the heart work, those cordyceps fungi that take over the brains and immune systems of insects, that's a low-hanging fruit. We know they're producing all kinds of compounds, but even the best collections have a handful of strains. And so we should be going to gather those strains that we know are likely to be in the value, and then we put them in that same pipeline. And so I think it's that putting those two things together. And in my experience, the people with the really big pipelines don't go outdoors very much. And so even if you think it should be obvious you should look there and add to your collection, it's not obvious. And so I think there's some real opportunities, and I'm starting to see them materialize in that context. But I'll stop there and I can come back to it later. Yeah, interest. But yeah, that's my answer. Thank you.
The implication I got from your talk is that our microbiome is pretty messed up because of our Western diet, living indoors, et cetera. So has there been any thought of going to a more pure society, let's say a Stone Age society in the Amazon or something, and talking them into giving us a fecal specimen and transplanting that to get back to a more pure microbiome?
Yeah, so there's actually a lot of work on this. And so, for example, the Hadza, hunter-gatherer group, the Hadza, have had their, Justin Sonnenberg, who I mentioned earlier, his group has worked on the Hadza to look at gut microbiomes and found that the Hadza have many of these microbes that seem to be missing from Western societies for the last couple hundred years. And so there now are attempts to figure out how do we think about using those microbes in fecal transplants? How do we think about selecting some of those microbes? As you might anticipate, the legal and ethical attributes of doing that are really complex. Who owns the gut microbiomes? Do those individual Hadza people Does the Hadza community? How do you think about that partnership? And so that work is happening. The other trick is, if you take one Western gut microbiome, so if you've got C. diff and you zap your gut microbiome to restore it, to get a fecal transplant, with another Western gut microbiome, it establishes pretty well typically. It's not clear that the same is true with the Hadza gut microbiome. And in part, probably the immune response to that very different gut microbiome is very complex. And so there's this added layer there. But for sure, there's a lot of work in this vein, and there are people kind of at the margins of science, but not so much at the margins, that are pushing for thinking about like a paleo gut microbiome. And you will see this more and more. But it's tricky for all these reasons. And I mean, if anybody interested in law, this is a legal area that will be rich for a long, long time. But there is some focus. Skin microbiome is the same. So there's studies of Yanomami, indigenous individuals in the Brazilian Amazon. One individual Yanomami boy who was sampled had more species of skin microbes than are known from the entire North American population. And so the changes are really extreme, and how do we think about that? But now there's a company selling those microbes, and you can buy it. Clinically, what's the value? We don't know yet, but yeah.
Just a question with the use of masks and more diligent hand washing over the last couple of years. Has there been any change that you see in disease processes or immunology of infectious disease in the last 2 years, other than just we're social distancing and we obviously don't have as much flu and colds and stuff? But ultimately, do you think in the future, is that a good thing to have the masks forever, or does? I mean, there's a lot of things.
It's tricky. I mean, this is the trade-off too. Like so I'll say first what we know. So there have been studies of frequent hand washing with antimicrobial compounds, alcohol or triclosan. And in those cases, you see persistent changes in the hand microbiome in some cases that look as though they're probably not great. And so collectively, depending on which substances we're using, we're probably triggering some shifts in our hands. And the first papers on that are just starting to come out. And in some cases, those are probably sort of pro-inflammatory changes. And so I would predict we see some more rosacea, some more of these inflammatory skin disorders in the next couple of years. But the COVID-specific effect is really, we're just seeing those papers. With the masks, there's this trade-off between acquiring the microbes you need and the value of keeping away the microbes we don't want. And it's really tricky to think about how you balance that trade-off. And it's different during a pandemic than it is outside of a pandemic. But probably there's less acquisition person to person of gut microbes during these last 2 years than there typically is. Mm-hmm. I would expect to see some effect of that, but it's probably very complex.
We were talking about this just as a group in a book club. And, most people go to the dentist and go years and years without dental decay. Over the last 2 years, a group of 12 women, 8 of us had cavities. Isn't that interesting? I don't know anything that could have caused that other than maybe we're colonizing with more facial or staph or something that wasn't normal mouth flora.
Yeah, and I've not talked about it, but the Western mouth flora is super different from the historic mouth flora. We actually have dental samples from Neanderthals and from ancient humans that we can get from teeth. And so we actually have a much more pro-cavity mouth flora than we've ever had before. And I don't know if it's changed during COVID, but it's clearly different from what we used to have. Yeah, but interesting questions.
Wondered if you had any comments about the whole genetically modified food industry and how GMOs are potentially affecting our biome.
Yeah, so it's a great question. So I was just in a big microbial foods meeting in Denmark last week, and so I'm a little more tired now than I should be. And in the European landscape, GMO foods are not allowed for the most part, but the products of GMO foods are. And so you can buy a microbial food that's made with GMO organisms as long as the microbes are gone. And so right now, the legal landscape for transgenics is really kind of, I mean, in some level, it's silly because most of the things people worry about are actually from the products, not the organisms. But what's happening on that landscape is the US sort of transgenic world and the European world are vastly different places. And so whereas the European world for microbes is really moving toward microbial therapeutics that take body microbes and give them back to us, the US model is really moving toward all these transgenic microbes. And my experience in looking in this agriculture is that there's not anything intrinsically better or worse than these transgenic organisms, but by their nature, they're new, and so they've typically not been studied how they work. And also, they're almost never studied in the context of other species. And if you have transgenic corn, that's one thing, because you're mostly thinking about pests and pathogens and corn microbes. And so it's a relatively simple system. When you're introducing transgenic microbes to the human body, you now have thousands of species interacting with those. And I don't think, it's hard to even know how we're going to get a good understanding of that landscape, but that work is moving very quickly. And in the US, the regulation is very loose. And so I think what that looks like for you clinically in the next couple of years is going to be, you're going to have people banging on your door to sell you these new transgenic organisms, and how do you think about them? And I don't think we're ready. The other piece is that in the agricultural context, most of the transgenic organisms are owned by 3 companies. And most of the science on them is owned by those companies. And so if that also happens with these microbes, then that's probably actually a bigger challenge than is the organisms themselves, is who controls them, how do we think about them, how do we think about all that protected science. And it looks like, at least on the food side of this, that's going to move in the same direction. Right now, for microbial foods, there are tens of companies that produce Impossible Burgers and all these different things. Most people predict that there are going to be 4 or 5 companies in the next years. And so they will own that technology, they'll own that knowledge, and how do we think about that? Yeah, great question.
Real quick.
Oh, sorry.
Yeah, you touched on one. I have 2 quick questions. One, when should we have you back? As fast as things are moving, when would you recommend? And then the second question, I was intrigued by the vaginal birth versus cesarean comment you made, and would you just expound upon that a little bit?
Yeah, so the work on vaginal birth really started relatively recently, and the earliest work tended to suggest that human babies were acquiring their gut microbiomes from their mother's vaginal communities. And so there was this vaginal community to gut community transfer. That paper I showed in my talk, and I'm glad to share it, actually adds some complexity to this and suggests that it's really kind of dose dependent, that which microbes the baby gets in its gut from vaginal birth are dependent on what it gets the biggest dose of. And so that most often in a Western context anyway, it tends to be fecal microbes from the mother during the messy process of birth that are establishing in the baby. And there are all kinds of hospital microbes that are arriving too, but they're outcompeted by the size of the dose. C-section babies seem to get most of their microbes from the hospital room and then from touching the individuals in the hospital room. And so in some cases, those C-section babies are getting really unusual microbes that have direct negative consequences. In other cases, they seem to still acquire their reasonable gut microbes, at least in a Western context. And we don't know yet what accounts for that variation from one baby to the next. Part of it may be that there's kind of a 2-year window when it's pretty easy for those babies to still acquire microbes. And it could be the messy process of just being in the house, and we do see some dog microbes being picked up in the C-section babies. But what we do know is that there are a whole suite of lifelong negative health consequences for the subset of the C-section babies that don't acquire a normal-ish gut microbe. And that's allergy and asthma, it's gut dysbiosis, and those studies are coming out very quickly. There are now certainly tens of them, and maybe 100 or more. And so if you're interested, I can point you to some papers. But obviously, this is a big deal in the context that a large proportion of babies in the US are now born C-section. And in many cases, it's medically necessary. And so how do we think about that? I'll tell you that my colleagues, when they have babies, almost always store feces so that if their babies don't get, or are born C-section or don't get the right microbes, they have some of their own feces that they can repopulate the baby's gut with. And they've been doing that for a decade now. And so if you go to a microbiologist's house, be careful in the freezer. And I'm glad to talk more about that later, but that, yeah.
I actually had two questions, and the first one was for C-section babies, how long was the time period that maybe you could repopulate? Like, would you recommend probiotics up to 2 years to try and help with that? So that was kind of my first question. And then my second question was, any studies about like indoor plants in households? Because my husband hates my indoor plants, but if I had more evidence to tell him it's good for us, that would be helpful.
For obvious reasons, the clinical studies of changing the exposures of those C-section babies to microbes are very few. With one quick exception. But in general, the thought is the first 2 years is when there's the most opportunity to acquire those microbes. It's not very clear, there are a handful of studies, what you might best do to make that happen. Microbiologists have decided that keeping some feces handy is the best way. Don't trust a microbiologist in everything. The other thing we know is in the 1960s, there was a team of skin microbiologists that noticed in hospitals that there was a Staphylococcus aureus that was becoming very common and causing skin infections in newborns. It's a longer story. It's in the Never Home Alone book. But basically, they also recognized that a subset of the babies in the same hospitals weren't picking up that Staph aureus. They noticed that those babies were being touched by the same nurse right after birth. They took staph from that nurse and started to inoculate newborn babies. And in doing so, they were able to establish that nurse's Staph aureus on those babies and rid those hospitals of the infectious Staph aureus. That approach spread to hospitals across the US. It was wildly successful. And then methicillin came on the market, and everybody switched to just using antibiotics and abandoned that approach of inoculating babies. And so we've been thinking about this for a long time. We've had approaches that work, but when you have antibiotics ready, often it's the easier thing to do. And so there's an interesting history to think about, too, that I think we're not super aware of. And hopefully I answered those questions. Like maybe I missed something. Oh, the plants. Thank you. Plants are tricky. It looks like the plant-associated leaf microbiomes are beneficial. Lots of work in Finland showing this. Finland's very different biologically. You could imagine reasons it would work in Finland, not other places. But if plants are watered a lot, there seems to be a tendency for them to favor bacteria that have antibiotic resistance. And so there's a trade-off between the soil and the plant and the leaves of the plant as a function of how much they're being watered. And there are like 3 studies on this. There's a super terrible old NASA study, and then there are a couple more recent ones. But I think if we could figure out how do we grow plants in ways that provide benefit, there's a lot of interest. And I'm actually working with architects now to think about how do you plant landscapes around low-income housing that bring the beneficial effects of biodiversity to families and kids, and how would we do that in a sensible way? But early days.
These were all wonderful questions. I'm pretty sure all of us are thinking about all the different conditions we treat and whether the microbiome is causing or related to it. So it'll be an interesting conversations to have here in the next few days. Just one question I had for you. It's a little more on the personal side. We talked about how kind of symbiotic our microbiome is with us. When you think about your own microbiome, do you think about it as being a part of you or more of a symbiotic partner with you?
Yeah, that's a great question. So we've worked for years on getting the public to think more sensibly about the value of their microbiomes. And I spend a lot of time working on this. Yet in the back of my head, I think about me and I think about them. I can't make that cognitive leap to think about the whole. What's interesting is that our immune systems think about the whole. Our subconscious immune systems are really doing all sorts of things to balance which of these species they're favoring and which ones they're disfavoring. And so there's this disconnect with what our body's doing and what our conscious minds are doing. The closest I can ever get to sort of appreciating like the whole is if you close your eyes, all the smells produced by somebody else are all smells that are body plus microbe. And sometimes good smells, sometimes bad smells, but it's this one moment in which you're experiencing them holistically. And so it's a way of remembering that there is this whole that our eyes sort of tell us is, our eyes keep telling us we're just a body, but our nose actually tells us we're something more complex. But per the question, I just nonetheless struggle to think about the whole. I nonetheless think about, me and them and this divided parts, because it's just a hard way to think, maybe especially in Western cultures. But thank you, and thank you everybody.
Thank you very much, Dr. Dunn.